Okay, so good morning. This is chapter 2, part 1 from Veterinary Immunology from the book of Ian Tizard. Okay, so name of the chapter is the invaders of the animal's body. So, any pathogenic organism, by the way, what is a pathogen? Any organism that can cause disease in an animal or human or any vertebrate that is called a pathogen. So, all the pathogens can be categorized into three categories: eukaryote, prokaryote, and viruses. Eukaryote, prokaryote, and viruses. So eukaryotes include fungi and protozoa, prokaryote include bacteria, and viruses include virus. Okay. Now, pathogen, any organism that can cause disease, and virulence, the ability of an organism to cause disease in terms of its severity. So there are two terms, virulence and pathogenicity. What is the meaning of virulence? Virulence means that the ability of an organism to cause disease in terms of its severity, and pathogenicity is a general term. Okay, it is a general term which refers to the ability of an organism to cause disease. Okay, simple. Okay. Next, we move on to pathogens are again of two types: primary pathogen, opportunistic pathogen. Okay. So primary pathogen, here the pathogen can invade a healthy individual even in low number. Okay. So primary pathogen means that the type of pathogen which when even present in low numbers, then their concentration is less, then also they can cause disease in a healthy animal. That are called primary pathogen. For example, Brucella abortus, canine distemper virus. This is canine distemper, by the way. CD. Okay. And opportunistic pathogen, here the pathogens can only cause disease under two conditions: when the immune system of the host is weak or pathogen is present in very large number. Okay. So these. Example, Pasteurella hemolytica. Okay. Next we'll move on to one of the most important terms of complete immunology, that is antigen. So two most important terms of immunology are antigen and antibody, as we know. So antigen, any component of pathogen which leads to the synthesis or production of antibodies. Antigen are any component of pathogen which leads to the production or synthesis of antibodies, that is called antigen. So we'll discuss different types of antigen. Microbial antigen, first is bacterial antigen. So now bacterial cell surface has four types of components on it, that is cell wall, capsule, pili and flagella. Okay, cell wall, capsule, pili and flagella. So if we talk about cell wall, then there are two types of cell wall: gram positive and gram negative. Gram negative, gram positive cell wall. So if we move on to gram negative cell wall, then here the cell wall is made up of polysaccharide dependent proteins. Okay, polysaccharide dependent protein, that is called lipopolysaccharide. And the antigenicity of the gram negative cell wall is linked to polysaccharide. Okay, the cell wall of lipopolysaccharide component are toxic, that's called endotoxin. These are the general points of general bacteriology, not of immunology. Okay, but they were given in Tizard, so I wrote them here. Gram positive cell wall, their cell wall is made up of peptidoglycan. Okay. Next we move on to capsules. Capsule can be of two nature: polysaccharide and protein, and polysaccharide are considered good antigens. Why good antigen? So what is the meaning of good antigen here? Good antigen means that they will lead to the mass production of antibody. They will lead to a production of antibody in a very large number. Okay. So why? Because they contain hexosamines. So that is why polysaccharide are considered good antigen. Okay. And hexosamine are better antigenic property. Have better antigenic property. So function of capsule, as we know, protect the bacteria against phagocytosis. Okay. Capsule antigens are called K antigens. So I have made a table here, this table. So we'll discuss this. But pili of fimbriae. So these are the short projections that cover the surfaces of some gram-negative bacteria. Pili antigen are called F antigen, and pili have role in attachment of the bacteria to host tissues and bacterial conjugation, bacterial reproduction. Okay. Now bacterial flagella have a role in motility. This point is extremely important. Any bacteria which is motile because of flagella. It consists of a single layer of protein called flagellin, and flagella antigens are called H antigens. Okay, so this is this. Okay. Now if we move on to this one, then this table is important. Cell wall antigen is O, capsule antigen is K, pili of fimbriae antigen is F, and flagella antigen is H. So these are important. Okay. Now other bacterial antigens are porins, heat shock proteins, and exotoxins. Okay. So the other bacterial antigen, despite of these, cell wall, capsule, pili of fimbriae, and flagella, that is OKFH respectively, they are porins. So porins, these are the proteins that forms the pores on the surface of gram-negative bacteria. Heat shock protein, protein generated in large amount in stressed bacteria. Exotoxin, these are the toxic products released by bacteria when they die. Simple. Okay. Now, exotoxins are highly immunogenic and they promote the production of antibodies. And in this case, the antibodies are called antitoxins. Simple. Now viral antigen, virus are obligate intracellular parasites. Obligate means they cannot survive without the host. Intracellular means found within a cell, single cell, and parasite means parasite. Okay. The components of viruses: capsid, capsomere, and nucleic acid core. So capsid, these are protein layers. Capsomere, these are subunit of the protein layer, and nucleic acid core. Okay. Okay. Now, some viruses might also have an envelope. An envelope is made up of lipoproteins and glycoproteins. What structure of virus act as antigen? Capsid and envelope, if present. If envelope is present, then it will also act as antigen, and capsid protein act as antigen always. A complete viral structure is called a virion. This is the topic of virology, not immunology. Okay. Now, immunopathogenesis. So, virus infects an animal cell. Protein of capsid acts as antigen, but the viruses are not always found in the blood. Dekho, what this is trying to say is virus will infect an animal cell, and that virus will enter into the animal cell. Okay, because they are obligate intracellular parasite. But the proteins of capsid act as an antigen. Proteins, as we have already discussed, that they act as antigens, but since these viruses are found inside the cell, not in the blood, so it is difficult for immune system to detect them and kill them. Okay, so what will happen here is, rather they are mostly found within the animal cell, but when the virus integrates the DNA RNA of its own with the cell's DNA, what it means to say is, when the virus will integrate its own DNA with the cell's DNA, then the animal cell will produce viral proteins, and these viral proteins are then transferred to the cell surface, host cell surface, and these then act as endogenous antigens, as we have already discussed in the first lecture on immunology. Okay. Now, non-microbial antigen: food particle, inhaled dust, snake bites, mosquito bite, through vaccine, through blood transfusion, experimental purposes, organ graft. These are different examples of non-microbial antigens. Cell surface antigen. The surface of every mammalian cell consists of hundreds of protein molecules, and most of these proteins act as antigen. Okay, blood group proteins found on RBC act as antigen. Example. Okay, so the most important protein that trigger graft rejection are MHC, that is major histocompatibility complex. MHC, what are MHC? These MHC molecules are found on surface of WBCs. So these MHCs are the most important type of protein molecule that trigger the graft rejection, that is rejection of the part of any transplantation and rejection of transplanted organ. Okay. Now, since each cell surface contains hundreds of proteins which act as antigen. Okay. Now, since each cell consists of hundreds of proteins which act as antigens, so a numbering system was assigned. Since now each of the cell has hundreds of antigens, proteins which will act as antigens, so numbering system was assigned. According to this numbering system, molecules with similar epitopes were grouped together. So, what is the meaning of epitopes? That we will discuss later in this part of lecture, but in this second part of this lecture. So, molecules with similar epitopes were grouped together. Okay, thus all monoclonal antibodies. So, what is monoclonal antibody? Antibodies produced against specific cell surface proteins. Okay, so these are called monoclonal antibodies, and so all the monoclonal antibodies that detected a single antigen are assigned to a number CD, that is cluster of differentiation. So these examples are important. CD4, CD8. Cluster of differentiation 4, cluster of differentiation 8. CD4, this protein is associated with the lymphocyte that help in immune response, and CD8 found on lymphocyte that kill the abnormal cell. Okay.